transcription. This is called transcription. So what is the definition of transcription? That is synthesis of RNA molecule by using of DNA as a template. Transfer of this information, because you already know DNA RNA is a genetic material and it is contains some genetic code which is responsible for different expressions of proteins in the body. Okay, so by using this DNA as a template, the result in the transfer of information is stored in double-stranded DNA into single-stranded RNA, which is used by the cell to direct synthesis of proteins. Okay, synthesis of protein. Now we can differentiate these transcription in two ways: transcription in prokaryotes, transcription in eukaryotes. Okay, so firstly we discuss transcription in prokaryotes. Now we understand all these topics in very well in the board. So this question, if the question is asking in your examination, it is asked in long answer questions, it is asked in short answer questions also. The question is asking what is transcription. First you give the expression of protein inside the cell. It is called transcription. Okay, so this is the definition. Here we require polymerase enzyme. Okay, ribonuclease polymerase enzyme. We discuss bacterial transcription here. What we discuss? Bacterial transcription here. Okay, it is a transcription of prokaryotes. And what is the requirement? Requirement for this transcription, requirement discuss one by one, and it is a very easy process. And there nucleotide triphosphate. Nucleotide triphosphate. What is basically what we do here? What we do here? In the DNA molecule, there is the presence of thymine. In the RNA molecule, there is the presence of uracil. Okay? So this is basic difference. This transcription, there are mainly three steps. The first one is initiation. Second one is elongation. And the third one is termination. Okay, this being the three steps. So if the question is asking write about the transcription of prokaryotes, you will write in this format. First you describe the transcription, then you will give the RNAPase. RNAPase is an enzyme in prokaryotes. There are basically difference between prokaryotes and eukaryotes. In eukaryotes, there are three basic classification of this RNA polymerase enzyme. RNA polymerase enzyme. Which one is that? RNA polymerase first, RNA polymerase second, and RNA polymerase third in eukaryote. We discussed in eukaryotes. But in prokaryotes, these are present in combined form. These are present in coordinate molecules forms. So firstly, it is RNA enzyme. RNA what? Can you tell me what is the meaning of holoenzyme? The enzyme present with core molecules and some prostatic groups. Okay. So hollow enzyme molecules, what is there? It contains enzyme. That means it contains alpha 1, beta, omega unit with a protein called sigma factor. Sigma protein. With a protein called sigma protein. Okay. How it is present? We call this is core enzyme, and with this hollow enzyme. We talk about RNAPase, RNA polymerase enzyme in prokaryotes. How it is present? It is present in alpha unit, alpha 1 alpha dash, beta 1 beta dash. This is alpha 1 alpha dash, beta 1 beta dash, and there is a present of omega. Okay, and here there is sigma proteins are present. So now what is the function of this particular separation of these enzymes? These sigma proteins scan the DNA template. Why this protein is scanning this DNA template? It is simply search a appropriate space in the DNA template for the attachment of RNA polymerase enzyme. Understand? Such like that, you are just walking in the ground, and when you want to take the lecture, you are coming in the lecture hall. So what we do? You will scanning the lecture hall to attend the lecture. So to initiate the transcription process, these sigma proteins scanning the DNA molecules and attach at an appropriate space to start transcription. You just easily understand it is scan the DNA molecules. What is this scan? What is it scan? It scans some particular sequences, some base pairing sequences. We call these sequences as Pribno box. You already listened the name. What is the meaning of Pribno box? That is Tata box. Okay, we understand all these things. Okay, what is the function of polymerization? Attachment of nucleotide triphosphate molecules with fresh DNA RNA templates. Okay, it do polymerization. And this alpha is used for opening of coil of DNA. How DNA molecules are present? If you read the molecular biology, you easily understand. It is present bind with histone proteins. Okay. So what is that? They firstly open this coiling. Okay. So these are the units, and all the subunits have different functions. Understand? RNAPase in the prokaryotes, they are present in combination form. We call this beta subunit, alpha subunit, omega subunit with sigma proteins. And all these particular subunits have different functions. Okay? So this is called RNAPase. Now we understand the template, promoters and NTP regions. Template. Template means what? Simply and this is now you understand the basic thing about that. We have two strands. The first one is template strand. I will ask the question to anyone. Be careful in my class. So this is called template strand. This is called template strand. You understand the terms. First you understand the terminology of this particular process and okay, template strand means what? What is the meaning of template strand? It is called self-strand. Negative. Okay. Now from the numbering of this base pair, we start our transcription process. So this is the template. What we required here? Template. We required here this template for the attachment of our RNA polymerase enzyme. Okay. Now we go understand all these things. Now what we required here? Nucleotide triphosphate, that is ATP, ATP, three types of nucleotide triphosphates are required. These three types of nucleotide triphosphates are required because here what we form? We form RNA molecule. That's why uridyl is present against thymine. Uracil triphosphate. Okay. Now promoter regions. What is the meaning of these promoter regions? You understand the terms. You will. Now we understand the term that plus one. This is plus one position and we call this transcription starting site. Okay. From plus one, if we go for this negative sign upstream, and this is the template which is used for the formation of RNA. All this stream negative sign, positive sign. Okay. Now from this one side, if we are going for next -10 position, minus 10 position, then there is some sequence of amino acid is present, and we call these sequences TAT. This is called Tata box. What we call Tata box, and it is called promoter region. What we call this? Promoter region. Promoter region. And then for next is TT. Sequence. Consequence. Sequence. Have you understand up to this? Have you understand up to this? What we are here? We just separate the DNA strand, double helix strand, and what is present in the DLA? There is base pairs are present. Okay? From one strand, we start the transcription side from +1, then it having some sequences, we call this Tata promoter region. This is promoter region, this again is promoter region. So I already tell you what the function of sigma protease. It scans the promoter region for the attachment of RNA polymerase enzyme. And we called this all these sequences, we called this as transcription unit. We called transcription unit. Understand? This is transcription unit. And the RNA which is formed after the pre-initiation complex, we understand all these things in the next step. So you understand that we have template, promoter regions, and nucleotides. We have some sequences. We call this sequence as promoter region second. Firstly, the sigma factor, sigma proteins scans the DNA molecule and it is attached with these promoter region. After then, it attached with Tata box. There is attachment of RNA polymerase enzyme. Okay? So this is called transcription unit. I think you understand all these things. Now we discuss one by one all the steps. Initiation, elongation, and termination. We understand all the steps. And okay, we discuss after. There is first step called close complex. Complex. Okay. Now the second step is open complex. Open complex. Now the third step is synthesis. Synthesis of 5 nucleotide base pairs. 5 to 10 nucleotides base pairs. Okay. And lastly, in the and clearance of promoter sequences. Clearance of promoter sequences. Now we understand all these terms very. this is the first. It is 3 prime to 5 prime. 3 prime to 5 prime. And we have +1 transcription starting sites. Okay, transcription starting site. After then -10 base pair. What is present? What is present? Very good. Tata box. And again there is some sequences are present. Please tell me the base pair of these sequences. Tell me base pair. T, TG, TG, T. Now, what is mainly here? What is the main? Why we called? Firstly, the sigma factor which is present in the RNA polymerase enzyme in prokaryote, it scans the Tata box, and here it is attached with that, and it is called close complex. Or another name of this close complex is pre-initiation complex. Pre-initiation complex. Okay? Pre-initiation complex. Now, in the next step, what is happening here? There is the formation of open complex. So open complex means what? There is bubble formation. What is formation here? Bubble formation. You see, in the next step, there is prime, and there is attachment of RNA polymerase enzyme, and here this is formation of bubbles, means it distorted the coiling of the DNA molecules and it forms bubble formations. Okay, so this condition is called open complex. Okay, now after then synthesis of 5 to 10 base pairs. Now what is happening here? There is the function of RNA polymerase enzyme. What is the meaning of polymerization? What is the meaning of polymerization? Attachment of nucleotide base pairs with 3' OH group of these newly synthesized RNA molecules. Newly synthesized RNA molecules. So what is there? 5 to 10 nucleotide base pair. 5 to 10 nucleotide base pair, or addition of 5 to 10 base pair for newly synthesized this RNA template. This being step third. How it is possible? How it is possible? Anyone can tell how it is possible addition of these base pairs from this newly synthesized template with the help of enzyme RNA polymerase. And what it contains in the RNA polymerase? It contains beta subunits. Beta subunits are responsible for polymerization of these nucleotide base pairs. Understand? Nucleotide base pairs. Okay? And after then, clearance of these promoter sequences. What is the meaning of clearance of these promoter sequences? Now these RNA polymerase enzyme change their position in the studied prime direction. Go to 3' direction, and due to this, they can change the position from these promoter sequences. And we called this as clearance of promoter sequences by RNA polymerase enzyme. Clearance of promoter sequences by RNA polymerase enzyme. Now repeat again all these initiation steps. Okay, you listen carefully. If you have any queries, you will ask. We have DNA templates. In the DNA templates, we have template and coding strings. Okay, in the template up to +1, there is transcription starting sites, and we going for upstream direction up to -10 base pairs. There is a presence of Tata box, and we call this Tata box as a promoter sequence. What we call promoter sequence. And if we going forward to same direction in -35 base pair, there is a presence of consequences sequences. What is the need of this promoter sequences, consequences sequences? It will helpful for the attachment of RNA polymerase to this DNA template. How it is attached? With the help of sigma protein. Sigma protein is scanning these DNA molecules. Understand? This sigma protein is scanning these DNA molecules, and it is firstly attached in consequences sequences, then it is attached with particular promoter sequences, which we call Privno box or Tata box. The another name of this promoter sequences is called Privno box. Sometimes question is asking, write about Privno box. You are not confusing here. Privno box is simply first promoter sequences. Now, in this step, in initiation step, there is first is formation of closed complex. Closed complex means what? Simply the RNA is attached, RNA polymerase is attached with the DNA fragments. RNA polymerase is attached with the DNA fragments. You easily see the diagram if you have any confusion. Simply you see these RNA polymerase enzyme recognize the promoter sequences and attach with that. Binding of RNA polymerase enzyme, and we called this step as a closed complex. What we call closed complex. Now, after the attachment of this RNA polymerase, these can unbind these DNA molecules and formation of bubble. You will see there is a formation of bubble, and we call as open complex. Open complex. And in this initiation transcription complex, there is now in the next step, there is elongation. There is elongation. Up to this, you all understand the meaning of initiation. Now we go for elongation. Don't talk. Trend. Okay. So in initiation, what here there is formation of 5 to 10 base pairs. Now in the next step, there is stretch of this newly formed RNA template by attachment of nucleotide base pairs. Okay. So this is called elongation. This is called elongation. Okay. Elongation process is very simple. You easily see there is nothing to worry. There is stretch of this RNA template, and there is elongation process occur. Elongation process occur. Okay. Now the last one is termination. Termination process in prokaryotes. In prokaryotes, it is to be simple. It is very simple. Okay. Okay. Now there is a special base pairing sequences which we call AAU, AA, and CA sequences are present. What is happening here? There is some, in the last step, there is cleavage factors are there. Some cleavage factors and some cleavage and polyadenylation. Polyadenylation complex. Polyadenylation complex. Polyadenylation complex and some proteins which having endonuclease activity. Endonuclease, the particular fragment internally. By this endonuclease activity, it can rebound these newly formed DNA from DNA. It rebound attach RNA complex. DNA RNA complex from this bubble, and we get separate RNA strand. Separate RNA strand. It is simply called transcription. With the help of some cleavage factors, some polyadenylation sequences, and some these endonuclease proteins, they will cut these fragments and separate out this RNA template and DNA template, and here our motto is over. So what is our motto here? Formation of newly RNA template. So you will see there is initiation, polymerase terminate, and release RNA. So after then, what is here? There is all the enzymes are separate out, and the RNA fragment which is newly formed is separate out from this DNA molecule. Okay? Understand? Understand the transcription in prokaryotes? I revise transcription in prokaryotes. What is there? There is simply template strand, promoter sequences, consequences sequences. Okay? So what does they do? They simply, there is attachment of RNA polymerase enzyme. With the attachment of RNA polymerase enzymes, it going for three-stage process called initiation, elongation and termination. In initiation, there is formation of closed complex, open complex, synthesis of 5 to 10 nucleotide base pairs, and lastly, clearance of promoter sequences. In the elongation, stretch of this RNA template, stretching of this RNA template by synthesizing, by addition of nucleotide base pair at C' OH group of this template, by polymerization, and lastly by some cleavage factor it is removed out. But these are separate in transcription in eukaryotes. Now we discuss transcription in mammalian cells or eukaryotes. Okay? So simply if the question is asking write about the transcription in eukaryotes, you also write it in this form. You also write it in this form. You will give the definition is same for eukaryotes and prokaryotes. Okay? And after that, there is again RNP enzyme, RNA polymerase enzyme. There is RNA polymerase second and third. These having separate RNA polymerase in eukaryotes, in mammalian cells. Okay? And there is a requirement. There is a RNAPase, some factors, and after then, same step. How many steps are there? Speak loudly. 3 steps. Which one? Initiation, elongation, and third one is initiation, elongation and termination. Okay? And we have 3 different enzymes, RNP first, RNP second and RNP fourth. So this and because RNA is having 3 types. Okay? So what is there? There is formation of first enzyme. Okay? Polymerase first enzyme. Okay, you learn these things. आपको याद होना चाहिए. There is formation of mRNA. And and RNA. Okay. All are the RNA polymerase first, second and third, which is used in the process of transcription in eukaryotes or mammalian cells. Okay. Now the factors are what? These are some special class of proteins. These are some special class of proteins. Which one? That is called, we divided these proteins as firstly as activator, secondly basal factors, and the third one is co-regulator. Third one is co-regulator. We divide these factors: basal second factors, co-regulators, and activators. Okay. Now we understand the example, that is the example of activators are second T, second B, second E, and lastly second H. All these are what? All these are basal transcription factors. Basal transcription factors. And lastly, co-regulators. And co-regulators are basically formed by transcription factor second D and having Tata binding proteins plus Tata binding proteins and 14 associated transcription factors. 14 associated transcription factors. Okay. So all these are the requirements for transcription process in the eukaryotic cell. In eukaryotic cell. Understand? Eukaryotic cell. Now we understand simply the step of this reaction. Step of this process. Step of this process. It is, if you make a diagram very with proper labeling. अभी एग्जामिनर क्या कर रहेगा? एग्जामिनर जो है उसको इतनी कॉपी चेक करनी है, 150 आपकी कॉपियां हैं, इतने क्वेश्चंस हैं। अभी आपकी कॉपी चेक करने बैठेगा, आप जनरल लैंग्वेज में पैराग्राफ में आप लिखेंगे तो क्या ही नंबर देगा आपको। अगर आप डायग्रामेटिक प्रेजेंटेशन देंगे, कुछ सब हेडिंग्स को बोल्ड लेटर में लिखेंगे और वो कॉपी के सामने इम्फेसिस होके वो चीज आएगी, तो आपको इजीली वो नंबर मिलेंगे। मिलेंगे कि नहीं मिलेंगे? यू विल गॉट इजीली नंबर। इफ योर प्रेजेंटेशन इज गुड, योर मार्किंग इज गुड। इफ योर प्रेजेंटेशन इज वर्स्ट, देयर इज नथिंग टू इम्प्रूव। कोई आपकी कॉपी सुधार नहीं आएगा। लिखना आपको है, किस तरीके से लिखना है, बेटर डायग्राम होना चाहिए, बेटर प्रेजेंटेशन होना चाहिए। ठीक है। नाउ देयर इज व्हाट, देयर इज transcription starting site. Okay, and if we are going for there is a presence of and it's moving. GC box. You understand this diagram? Why I am calling upstream, which one calling downstream? Upstream means moving towards direction from starting point. From starting point. Okay? In eukaryotic cell, there is the presence of these sequences which we call Tata box, cat box at particular region. Okay? So there is firstly the initiation, there is the form, and we call this unit as transcription unit. I already tell you in transcription unit. So in this step, there is again simple the step of initiation in prokaryotes and eukaryotes are same. Which one is the step? The first step is formation of closed complex, then open complex, then synthesis of 5 to 10 nucleotide base pair and clearance of promoter region. Okay. Now here there is something different. Different means was Tata binding proteins are there which recognizes Tata box. It is recognized by Tata binding protein, and with that some these transcription factors which attachment of polymerase enzyme with polymerase enzyme, then which we call helicase enzyme. So what is the function of this helicase enzyme? What is the function of this helicase enzyme? Unbinding of DNA coiling structure. Which enzyme is doing this unbinding of these DNA molecules? In DNA replication, this is topoisomerase. Topoisomerase. Okay. So all are associated with that. We understand one by one. We understand one by one. So firstly, there is the attachment protein, and this transcription factor D is what they do. They can bind this DNA fragment. Bind this DNA fragment. Students understand this. It binds the DNA. So firstly, this is called Tata box. So firstly, there is attachment of Tata binding protein. So this is your and after then what I tell you, there is attachment of transcription factor second D. Transcription factor. And again, there is the attachment of transcription factor A. Transcription factor F. This is transcription factor F. Here. Okay. This is called initiation complex. We call this as close complex. We call this as close complex. What is happening here? Firstly, there is attachment of tita binding protein in tita boss sequences. After then, there is attachment of transcription factor D, which binds the DNA molecules. After then, there is attachment of transcription factor A and transcription factor V with transcription factor F with polymerase enzyme. With polymerase enzyme. Okay? So this is called close complex. This is called close. Now in the next step, open complexes. It is same as prokaryotes. Formation of open complexes. Here polymerase enzyme is there with transcription factor F. Okay. There is retachment of this nucleotide base pair. Okay. Nucleotide base pairs. Nucleotide base pairs. Okay. So this is called formation of pre-initiation complex. Formation of pre-initiation complex, and we will go for in the next step. Don't talk. आवाज नहीं आनी चाहिए. I think time is over. So on Monday we discuss step of this transcription of eukaryotes, elongation process, and lastly termination, because both the processes are very important and it is separate from prokaryotes. Okay, because your time is next आपका DH का क्लास हुआ, प्रैक्टिकल्स का क्लास हुआ, ठीक है. मंडे में हम डिस्कस करेंगे ये दोनों स्टेप, एंड आफ्टर देन वी डिस्कस पोस्ट ट्रांसक्रिप्शन मॉडिफिकेशंस with their inhibitors. Okay, in detail. I think all of you understand this. Okay, we will also repeat transcription of eukaryotes on Monday. So all of you read this topic and come on Monday. We discuss in detail. Thank you.
And this medial border, inferomedial border, it is also divided. Anterior part is known as medial orbital border and posterior part is known as medial occipital border. Okay, understood? Now we have separated, and this is a temporal pole. You can say this is lateral view of brain. We have separated the frontal lobe from the temporal lobe, and in the deeper plane lies the insula or insular lobe, insular cortex, also known as island of Rayleigh. Okay, this can sometimes asked in your viva. Another, which lobe is present? Fifth lobe is, name the fifth lobe. Or island of Rayleigh can also come as your short note. Okay, and it is surrounded by this circular sulcus, and on the superior surface of this superior temporal gyri, there are 4 gyri are present, 2 small gyri, 2 long gyri. Small gyri are known as gyra brevia, long gyri are the gyra longa. Okay, so these are the, this is about insula. Now we are dividing the brain into 4 main lobes. Okay, by these sulcuses, important sulcuses. First is central sulcus. Here is central sulcus. This is lateral sulcus and its posterior rami, and extend it posteriorly first imaginary line. And here from the preoccipital notch to parieto-occipital sulcus, this is the first imaginary line. Here is second imaginary line. So all these lines together dividing the brain into 4 main lobes: frontal lobe, parietal lobe, temporal lobe, occipital lobe. Okay, understood? So in this view you can see all the 3 lobes, poles, occipital pole, frontal pole and temporal pole. Okay. Now we will discuss the main sulci and gyri on the superolateral surface. Okay. So first main sulci is the central sulcus. Central sulcus starts from the superomedial border 1 cm behind the midpoint between the frontal and occipital pole. Okay, from here it is starting, extending downward and forward and meeting this lateral sulcus. Okay, lateral sulcus, it has 3 rami: anterior horizontal, anterior ascending and posterior ramus. Okay, this anterior, okay, now we go from here, central sulcus, and parallel to it, anterior to the central sulcus is precentral sulcus, posterior to the central sulcus is postcentral sulcus. So in between central sulcus and precentral sulcus, here will be the precentral gyrus, which is the motor cortex. Okay, area 4 of Brodmann. And postcentral gyri lying between the central sulcus and postcentral sulcus, this is primary sensory area, Brodmann area 312. Okay. Now in the frontal lobe, 2 sulci are dividing the frontal lobe into 3 gyri from above downward. Superior and inferior frontal sulcus dividing the frontal lobe into superior, middle and inferior frontal gyri. Now this inferior frontal gyri, this is again divided by this rami of the lateral sulcus into 3 parts. This is anterior horizontal, this is anterior ascending. These are dividing the inferior frontal gyri into 3 parts: pars anterior, pars triangularis and pars opercularis. A, B and C representing pars orbitalis, triangularis and opercularis. Operculum means a lid. Okay, it is covering the insular cortex. This is frontal operculum and below there is temporal operculum. Okay, they are together covering the insular cortex from the surface. Now we go to the parietal lobe. Below this posterior to this postcentral sulcus, the rest of the area, there is only one sulcus, intraparietal sulcus. which is dividing the parietal lobe in the upper part superior parietal lobule, in the lower part inferior parietal lobule. Okay. Now we go to the temporal lobe. Here also two sulci are seen, superior temporal sulcus, inferior temporal sulcus, dividing the temporal lobe into three gyri, superior, middle and inferior temporal gyri. Okay, understood? Now this upturned end of the posterior ramus of lateral sulcus, around which one gyri is present, that is known as supramarginal gyri. This is area 39. Okay, supramarginal gyri is area 39. Next, around the upturned end of this superior temporal sulcus, this area is known as angular gyrus. This is area 40. Okay, these are important association areas 39 and 40, and these parts, this supramarginal gyri and angular gyri, these are located within inferior parietal lobule. Okay, understood? Next we go to the occipital lobe. Here there is extension of the postcalcarine sulcus. Around it, this U-shaped sulcus is the lunate sulcus, and here is lateral occipital sulcus, and here will be the transverse occipital sulcus. So these are the main sulci and gyri in the superolateral surface. You must know this central sulcus, precentral, postcentral sulcus, lateral sulcus is the three rami, you must know. Okay. Next we go to the medial surface. How will you identify it is medial surface? You can see the section of corpus luteum. Okay. What are the four parts of the corpus luteum? From anterior to posterior, there will be rostrum, genu, then body and splenium. Okay. So these are the four parts of the corpus luteum. Now see the important sulci and gyri of this area. Just above the corpus callosum, there is callosal sulcus. Okay. Parallel to the corpus callosum, above is the cingulate sulcus. Okay. In between the callosal sulcus and cingulate sulcus, here will be the cingulate gyri. Now here there is extension of the central sulcus here in this part, and around which anteriorly extension of the primary motor area, posteriorly extension of the primary sensory area. These two parts are together forming the paracentral lobule. Very, very important. Around the extension of the central sulcus on the medial surface, the area of brain is known as paracentral lobule. Very frequently asked in your viva. This is area for maturation and defecation. Okay. Now anteriorly, this part is forming the medial frontal gyri. Frontal gyri. Okay. Now posteriorly, here is one sulcus just above the splenium, that is known as suprasplenial sulcus. Okay. And this part is known as precuneus. Now this sulcus on the medial surface separating the parietal lobe from the occipital lobe, this is known as parieto-occipital sulcus, and this is known as calcarine sulcus, and its posterior extension is known as postcalcarine sulcus. So the part in between the parieto-occipital sulcus and the calcarine sulcus, this triangular part is known as the cuneus. Okay, understood? Next, we go to this temporal lobe. In this part, it is also divided by two sulci. One is collateral sulcus, another is occipitotemporal sulcus, dividing into three gyri. Parahippocampal gyri, the medial to this collateral sulcus lies this parahippocampal gyri. Then here is medial occipitotemporal gyri. Here is lateral occipitotemporal gyri. Okay, and anterior extension of this collateral sulcus. Here small sulcus is present. This is known as rhinal sulcus. Rhinals mean related to nose. So olfactory area is located here. This anterior projecting part of this parahippocampal gyri, this is known as uncus. This first is very important. You must know the uncus. You have to show it in your viscera. Okay, so uncus, here lies the olfactory area, area 28. Okay, and in front of this rostrum, here lies the paraterminal gyrus. These are very small gyri, and another is parolfactory gyri. Next, we go to the inferior surface. Inferior surface is divided by this lateral sulcus into posterior part is known as the tentorial surface, anterior part is orbital surface. Orbital surface, over the orbital surface, just parallel to the medial border, one straight sulcus is present. That is known as olfactory sulcus. Okay, why this name is given? Olfactory bulb, olfactory tract are lying along this sulcus. Okay, that's why olfactory sulcus. And one straight gyri is present just medial to this olfactory sulcus, that is known as gyrus recti. Okay, and rest of the part of the orbital surface, here one eight-sepate sulcus is present, that is known as orbital sulcus, and it is dividing the orbital surface into four gyri: anterior, posterior, median and lateral orbital gyri. Okay, understood? Next we go to the tentorial surface. Here you can see the extension of calcarine sulcus, postcalcarine sulcus, lunate sulcus, and these are the collateral sulcus, rhinal sulcus, and here is the occipitotemporal sulcus. And the three gyri, parahippocampal gyri, anterior extension is uncus, here is the medial, and here is lateral occipitotemporal gyri, extending into occipital lobe and temporal lobe. That's why the names are given. Okay, so these are the important sulci and gyri of the inferior surface of the brain. Understood? So now we will discuss, go to the Brodmann's area. Okay, functional areas of the brain. So cerebral cortex is demarcated into large number of areas which differ from each other in structure as well as in function. Brodmann in 2009 had divided the cerebral cortex into 47 such areas and indicated each of them by a number. Okay, so these are the different Brodmann's area. On the superolateral view, you can see this is central sulcus. Precentral gyri is area 4, that is primary motor area. Postcentral gyri is primary sensory area. This is 312. Then here is the premotor area, area 6. Here is the frontal eye field, area 8, and prefrontal area is 9 to 12. Okay, 9, 10, 11 and 12. Next, in the pars triangularis, area 45, and pars opercularis is area 44. These two are together forming the motor speech area of Broca. Very important. Motor speech area of Broca. Next, posteriorly in the parietal lobe, you can see area 5 and 7. These are secondary sensory area and or sensory association areas. Okay. Now, around the upturned end of this posterior ramus of lateral sulcus, here is area 40 in the supramarginal gyri. Now, upturned end of this superior temporal sulcus, there is area 39. This is known as sensory speech area or Oendix area, that is mainly area 39. And in the posterior part of this superior temporal gyri, here is lying area 22. Mainly this area 22 is known as Wernicke's area, and these three areas are together forming the sensory speech area. Area 22, it is coordinating with sound, auditory impulse, 39 is getting the visual impulse, and 40 is getting different sensory impulse, touch, pain, temperature, pressure, like this. So these three are together coordinating and they are together forming the sensory speech area. Understood? And primary auditory area is area 41 and 42. They are lying on the superior temporal gyri, mainly on the superior surface of the superior temporal gyri. Here is 41 and around it will be the 42. Okay. So they are mainly, you have seen on this during the insular cortex, gyra longa and gyra brevia, those parts are the auditory area, primary auditory area. Okay, on the superior surface of the superior temporal gyri. Location is important. Now in the occipital region, around the calcarine sulcus, here is area 17 or striate area or primary visual area. Next, around 17, there is area 18, that is known as parastriate area. Okay, that is visual association area, and around 18 will lie the area 19, that is higher visual association area or peristriate area. Okay, understood? So these areas are very, very important. Motor speech area of Broca, sensory speech area, that is Wernicke's area, containing area 22 mainly, also 39 and 40. This primary motor area, primary sensory area, and here this visual areas, area 17, 18, 19. Okay, these are very, very important functional areas. Next we go to the medial surface. On the medial surface you can see uncus and parahippocampal gyrus. Here lies the primary olfactory area. Okay, here lies within the cingulate gyri 23 and 24, and in the paracentral lobule forming part of the motor area, part of the sensory area is lying. They are together forming the paracentral lobule, center for micturition and defecation. Here is the premotor area, extension of area 6, and here is 8, 9, 10, 11, also 12 is present. Okay, prefrontal gyrus. Here on this medial surface you can see the three visual areas. Around the calcarine sulcus there is area 17 striate area, 18 is parastriate, 19 is peristriate. Okay, primary visual area, visual association area, higher visual association area. Okay, so these are important. So three main types of functional areas are there: motor area, sensory area, and association areas. Primary, now we will discuss the motor areas. First is primary motor area, that is area 4. It is located in precentral gyrus in superolateral surface, and motor homunculus is present over here. Okay, this is representation of different areas. How the motor function is more developed, that area is more enlarged in the cortex. So on the medial surface lying the foot and lower limb, then hip, trunk, then on the superolateral surface from above downward you can see representation of the hip, then trunk. Okay, then there is the arm, forearm, then hand area is larger because very precise movements occurs in the hand. Face also precise movements occur, different facial expression, movement of tongue or during speech. Here is the tongue and larynx. So these are the representation of sensory areas in the sensory cortex. Okay, that is known as, motor areas in motor cortex, that is known as motor homunculus, and they control the voluntary movements of the contralateral side. If paralysis occurs in the left hemisphere, right side of the body will be paralyzed. Okay, like this, vice versa. Now effect of lesion of the primary motor area: contralateral flaccid paralysis initially, decreased deep tendon reflex, Babinski sign positive, and moderate recovery of function occurs because it is upper motor neuron type of paralysis. Okay, in lower motor neuron type of paralysis there will be complete paralysis, no recovery. Next we go to premotor area, that is area 6 and 8. Here this is primary motor area, here is 6 and 8. Area 6 responsible for skillful acts, programming of movements. Upper part of area 6 is the writing area, Area 8 is also known as frontal eye field, regulates the voluntary conjugate movements. Next we go to supplementary motor area, continuation of area 6 on the medial surface. Here this is supplementary motor area. It controls the complex movements, elicit and urge to move. Lesions of this area will lead to akinesia, loss of movement, and upper motor neuron type of paralysis. In this case, there will be conjoint lesion of primary motor, premotor and supplementary motor areas. All these areas are paralyzed. Okay. Now, prefrontal area, that is area 9 to 12, 9, 10, 11 and 12. In front of premotor area, it is 9, premotor area was 6 and 8. They are highly developed in human, prefrontal area. These functions are very frequently asked in your viva of prefrontal area. Mainly all complicated tasks, higher thinking activities, all those are performed in this frontal lobe. Okay. So it regulates the depth of feeling, abstract thinking, mature judgment, foresight and tactfulness. This is the silent area of the brain. Okay. Next we go to the sensory areas. So we have covered the motor areas. Now going to the sensory area. There are primary sensory area, secondary sensory area only for somesthetic senses, sensory association area and higher association areas. Primary sensory area, these are again of three types: somesthetic area, visual area, auditory area. Primary somesthetic area, it is located in the post-central gyrus on the superolateral surface. Here this is area 312, that is primary sensory area or primary somesthetic area. What is the function? It localizes, analyzes, discriminates different modalities of cutaneous sensation like touch, pressure, position, vibration senses. Sensory homunculus are present here. Sensations are somatotopically arranged. Here also you can see on the medial surface there is foot and lower limb, then there is on the superolateral surface trunk, head and neck, then arm, forearm, then hand, face, like this. Okay. So this is the representation of sensory homunculus in the sensory cortex. Lower part of post-central gyri is the taste center, that is area 43. Okay. Here post-central gyrus lower part in this region will be area 43, that is taste area. Next we go to the primary somesthetic area functions. One important part is paracentral lobule, that is area for micturition and defecation, lying on the medial surface, receives senses of distension of bladder and rectum. Okay. Areas are 4 আর 312 দুটোই থাকবে। Next we go to secondary somesthetic area or supplementary sensory area. Upper lip of the posterior ramus of lateral sulcus, lower part of pre and post central gyri. This is the location of secondary sensory area. Functions: it receives the pain sensation and other modalities of cutaneous sensations. Okay. Next, sensory association area, that is area 5 and 7. Here area 5 and 7 lying in the parietal lobule, superior parietal lobule mainly, behind the postcentral gyri, superior parietal lobule location, area 5 and 7. Somesthetic association area. Next, higher association area for somesthetic senses. Somesthetic sense means touch, pressure, vibration, like this. Okay. So higher association areas are present in the supramarginal gyrus, area 40, and richly connected with area 5 and 7. Lesions of this area 40 will lead to tactile agnosia or astereognosis. Okay, astereognosis means you cannot understand and feel by touching the object if you close your eyes. Okay, and tactile aphasia. Now olfactory area, Brodmann's silence, কি কথা বলছিস এত আগে শুনে নে তারপর নিজেরা ডিসকাস করিস olfactory area is area 28 is located in the anterior part of parahippocampal gyri and uncus here primary olfactory area 28 uncus and anterior part of parahippocampal gyri next is visual senses primary visual area 17 you have seen visual association area 18 and 19 higher visual association area is area 39 here okay in the angular gyri now visual association area 18 and 19 parastite area is area 18 also known as visual area 2 and peristite area is area 19 also known as visual area 3 what are the function these are known as occipital eye field area 18 and 19 and mediate conjugate movements involuntarily they helps in conjugate movement of the eye conjugate movement means both eyeball will move together okay next is auditory area auditory senses primary auditory area is area 49 auditory association area is area 42 and higher auditory association area is area 22 here you can see 41 42 22 okay in the superior temporal gyri psychical cortex anterior part of temporal lobe is known as temporal pole here temporal pole this is known as psychical cortex what is the function it helps in memory present and past memory dreams hallucinations auditory and visual hallucinations occur in this area there is a circuit formation here okay from hippocampus parahippocampus amygdaloid body then this on the medial surface cingulate gyrus all these are together forming a circle C-shaped circuit like structure those helps in memory okay now speech is very important it is performed by left cerebral hemisphere regardless of the hand preference hence left hemisphere is dominant or talking brain and right hemisphere is non-dominant or mute brain now dominant hemisphere has 4 speech centers 3 sensory and 1 motor speech center sensory speech areas are these areas area 22 is higher auditory association area this is known as Wernicke's area area 39 higher visual association area and area 42 higher association area for somesthetic senses okay so amra kokhon we generally talk after reading or after auditory hearing okay so all those senses are together associated in these higher areas after that we can talk after understanding all those things visual or okay so all these things these areas are together forming the sensory speech area next is motor speech area is located in area 44 and 45 pars triangularis pars opercularis Broca's area very important so speech centers this motor and sensory may be transferred to the right hemisphere in child up to 6 to 8 years of age in case of cerebral injury okay generally it lies in the left hemisphere in both right and left handed person but in case of child if injury occurs on the left hemisphere the speech center can be transferred to the right hemisphere okay now this area 22 39 40 they receives input from hearing vision touch and proprioception 40 te chilo touch and proprioception 39 a vision 22 te hearing okay all these senses are associated here and so information processed in area 22 posterior part and projected to Broca's area through arcuate fasciculus arcuate fasciculus is connecting sensory and motor speech area see here the green area is the sensory speech area 22 39 40 and this is motor speech area Broca's area 42 44 and 45 This you are connected by arcuate fasciculus. কোথায় গেছলি তুই? ছুটি হয়ে গেছে যা। Okay, so lessons of speech area, lessons involving speech area, speech center or dominant hemisphere, it will lead to aphasia. Two types of aphasia is present, sensory aphasia, motor aphasia. Sensory aphasia, sensory speech area is destroyed, sensory aphasia, patient cannot understand anything. Okay, motor speech area, if motor speech area is destroyed, there will be motor aphasia, patient cannot talk but patient can understand. Okay, and conduct aphasia, lesion of arcuate fasciculus, connection between sensory and motor area. There will be fluent when a patient can talk and also patient can understand, but there is no correlation between understanding and talking. Okay, so there is fluent, patient is fluent, comprehension is also intact, and repetition of spoken language is extremely difficult in this conduct aphasia. So sensory speech area is located in the left dominant hemisphere, occupying the posterior part of superior temporal gyri of the temporal lobe, angular gyri area 39 and supramarginal area 40 of the inferior parietal lobule. And the Wernicke's area is concerned with the interpretation of language through visual and auditory input, and it is also an essential zone for constant availability of learned word patterns. The angular area or area 39 and supramarginal gyri are essential for process of learning. Okay, they are higher association areas, so learning is processed here. Now summarization, we have three types of areas mainly. Motor areas mainly in the precentral gyri, sensory area, postcentral gyri, psychical area at the temporal pole. Okay, motor areas are primary motor area, area 4 premotor area, 6 and 8, supplementary motor area are prefrontal area. Supplementary motor area extension of area 6 on the medial surface, our prefrontal area was 9 to 12. Okay, so these are the motor areas. Next is sensory area, primary sensory area 312, secondary sensory area only for somesthetic senses 5 and 7. Sensory association area is say area 5 and 7 lower part. Higher association area 40. Okay. Now primary sensory areas are somesthetic area. Somesthetic area is area 312, visual area 17, 18 and 19, auditory area 41, 42 and 22 is higher auditory association. Okay, so these areas are important. All those you have to see. Primary motor area, first the central sulcus, primary motor area 4, primary sensory area 312, motor speech area, Broca's area 44, 45 in the pars triangularis, opercularis, and sensory speech area 39, 40, 39 is supramarginal gyri.